Welcome back to the deep dive. Today, we are getting right into the biological wiring, the absolute foundations of, well, everything you think, feel, and do.
Yeah, we're talking about the nervous system. Think of this as the ultimate owner's manual.
Exactly. We're trying to give you the shortcuts, the core mechanisms. We're
Mhm.
talking neurons, brain structures, chemicals, genes, the whole blueprint.
Right. And understanding this biology is, well, it's key. It's the foundation for pretty much all the psychological concepts we talk about. We'll hit the essential definitions, sure, but also some, surprising facts, going from the tiny stuff up to the big picture.
Okay, let's get granular then. Let's start right at the bottom, the basic building block, the neuron.
Right. The neuron. That's the specialized cell, the one doing the signaling, transmitting information electrically and chemically.
The star of the show. But it doesn't work alone, does it? There are other cells involved.
Absolutely. You've got the glial cells. They're often overlooked, but they're critically important, kind of like the support staff.
I always thought of them as just like cleaning up waste products. Is it more than that?
Oh, much more. They do handle waste, yeah, but they also provide structural support, maintain the chemical environment around the neurons, and, crucially, many of them produce the myelin sheath.
Ah, the insulation. We'll
Yeah.
definitely get to that. So they're like the road crew and the maintenance team for the neurons.
That's a great way to put it, making sure the conditions are perfect for the neurons to fire properly.
Okay, so with that support structure in place, let's talk about the neurons themselves. There are different types, right? Based on their job.
Exactly. Three main types, defining the flow of information. First up, you have the sensory neurons. These are also called afferent neurons.
Afferent, meaning they carry signals towards the center.
Precisely. They transmit information to the spinal cord and brain. They're picking up signals from your senses, sight, sound, touch, but also from your internal organs and muscles, reporting what's happening out there and in here.
So touching a hot stove sensory neurons are the first responders yelling, danger, back to HQ.
You got it.
Yeah.
Then you have the motor neurons. These carry the commands from the central nervous system, the brain and spinal cord, out to the muscles and glands.
So HQ receives the hot stove message, and the motor neurons carry the command, pull your hand back now.
Exactly. They
Yep.
execute the action. And linking these two,
There must be something in the middle doing the processing, right?
right, those are the interneurons. They transmit information between other neurons. They're the vast majority of neurons, actually, especially in the brain and spinal cord. They handle all the complex stuff, the analysis, the decision-making, connecting sensory input to motor output.
So they're the analysts, the middle managers processing all the data. Okay. Hashtag the importance of insulation and signal speed. Now, for this system to work effectively, especially for quick reactions, the signals need to travel incredibly fast. And that speed depends a lot on the neuron's structure, specifically that insulation you mentioned, the myelin sheath.
Yes, the myelin sheath is critical. It's a fatty layer, mostly made by certain glial cells, that wraps around the axon, the long cable part of the neuron. Its main job, speed. It dramatically increases how fast that electrical nerve impulse travels.
It's like the rubber coating on an electrical wire, preventing leaks and speeding things up.
A very good analogy. But it's not a continuous coating, which is interesting.
Right, I remember reading about gaps, nodes of Ranvier.
Exactly. The nodes of Ranvier, these are tiny, uninsulated gaps along the axon between the segments of myelin.
Why the gaps? It seems like you'd want full insulation.
It seems counterintuitive, but the gaps are key for speed. It forces the electrical signal to sort of jump from one node to the next. This process is called saltatory conduction.
Saltatory, like salt.
No, from the Latin saltare, meaning to leap or to jump.
Ah, okay, so the signal literally leaps across the insulated parts, hitting the gaps.
Precisely. And that jumping is much, much faster than having the signal travel smoothly down an unmyelinated axon. It's a brilliant optimization.
And we really see how crucial this myelin is when something goes wrong with it, like in multiple sclerosis.
Absolutely. Multiple sclerosis, MS, is a tragic example. It's an autoimmune disease where the body's own immune system attacks and destroys the myelin sheath in the central nervous system.
So the insulation gets damaged.
Right. When that happens, the signal transmission slows way down or gets weak or might even fail completely. This leads to all sorts of neurological problems, vision issues, muscle weakness, coordination problems, fatigue. It really drives home how vital that fatty sheath is for normal function. Hashtag how the electrical signal fires.
Okay, so we've got the structure, the insulation for speed. Now, the signal itself, how does a neuron actually fire? It starts from a resting state, right?
Correct. Before it fires, the neuron is in its resting potential. There's an electrical difference across its cell membrane. Think of it like a tiny battery. The inside of the cell is negatively charged compared to the outside.
How negative we're talking?
Usually around negative 70 millivolts. This negative charge is maintained by controlling the flow of charged particles, ions, across the membrane. It's holding potential energy ready to go.
Like a drawn bowstring.
Exactly. Now, when the neuron receives enough stimulation from other neurons' signals arriving at its dendrites and cell body, it reaches a threshold. This triggers a sudden change.
What happens then?
Special gates in the axon membrane, sodium gates, fly open. And positively charged sodium ions, which are concentrated outside the cell, rush into the neuron.
So a flood of positive charge comes in. That must flip the internal charge, right?
Instantly. This rapid influx of positive ions is called depolarization. The inside of the cell membrane quickly goes from being negative 70 milliV, to becoming positive.
And that's the signal.
That abrupt change, that spike from negative to positive charge, is the action potential. That's the nerve impulse, the electrical signal that shoots down the axon.
And it's an all or nothing thing, isn't it? It either fires fully or not at all.
Absolutely. Once that threshold is reached, the action potential fires with the same intensity every single time. It doesn't get bigger for a stronger stimulus.
So how does the neuron signal intensity then if the zap is always the same size?
By the frequency of firing. A stronger stimulus makes the neuron fire action potentials more rapidly, more times per second. It's like Morse code, not louder dots and dashes, just faster ones.
Okay, so these action potentials are zipping along individual neurons. Now let's zoom out to the whole network, the nervous system. Our sources divide it pretty clearly into two main parts, right? Based on location.
That's the main division. You have the central nervous system, or CNS. That's the command center.
Brain and spinal cord.
Brain and spinal cord. That's it. It processes information and sends out commands. Then everything else, all the nerves branching out from the CNS, is the peripheral nervous system, the PNS.
Peripheral, like on the edges.
Exactly. The PNS connects the CNS to the rest of the body. Your sensory organs, your muscles, your glands. It's the communication link between the command center and the outside world and your internal body parts. It carries the sensory info in and the motor commands out. #tag divisions of the peripheral nervous system.
Okay. CNS and PNS. Now, within the PNS, that peripheral network, there are further divisions based on what they control, right? Voluntary versus involuntary.
Correct. First, there's the somatic nervous system. Soma means body. This part controls voluntary movements and transmits sensory information you're consciously aware of.
So deciding to pick up this pen, wave my hand, that's the somatic system relaying messages between my brain and my skeletal muscles.
Precisely. You consciously control it. The other major division of the PNS is the autonomic nervous system, or ANS.
Autonomic, like automatic.
Exactly. This system regulates all the bodily functions that happen automatically, involuntarily. Things you don't consciously manage.
Like breathing, heart rate, digestion.
Digestion, sweating, pupil dilation, all those background processes keeping you alive and functioning smoothly. #tag the ANS balance. Fight, flight versus rest, digest.
And the autonomic system itself has two branches that seem to work in opposition, creating a balance. This is the famous fight or flight stuff, right?
That's right. The ANS is divided into the sympathetic nervous system and the parasympathetic nervous system. They generally have opposite effects, working together to help the body adapt to changing demands.
Okay, let's start with the sympathetic. That's the fight or flight one, the accelerator.
Exactly. The sympathetic system mobilizes the body's resources for action, especially during times of stress, threat, or intense physical activity. Think of it as the emergency response system.
What does it actually do to the body when it kicks in?
It triggers a whole cascade of changes. Heart rate speeds up, blood pressure rises, breathing becomes faster and deeper, all to get more oxygen to your muscles. Blood flow gets redirected away from organs like the digestive system and towards the muscles needed for action.
Stops digestion because that's not important if you're running from a tiger.
Pretty much. It also stimulates the adrenal glands to release stress hormones like adrenaline, which further boosts arousal and energy mobilization. It prepares you to either fight the threat or flee from it.
Okay, so that's the go system. What about the other side, the parasympathetic?
The parasympathetic nervous system is the rest and digest system. It does the opposite. It calms the body down, conserving energy and promoting processes that restore resources.
So it slows the heart rate back down?
Yes, it slows the heart rate, lowers blood pressure, slows breathing. It stimulates digestion again, diverting blood flow back to the gut. It basically helps the body recover and replenish its energy stores after the stress or exertion has passed.
So they're like a gas pedal and a brake, constantly adjusting to keep the body in a stable state.
That's a perfect way to think about it. This dynamic balance between the sympathetic activation in parasympathetic calming systems is crucial for maintaining homeostasis, that stable internal environment your body needs to function optimally. Problems arise when one system is chronically overactive, like constant stress, keeping the sympathetic system engaged.
All right, let's loop back to the connection between neurons. We talked about the electrical signal, the action potential, racing down the axon, but it stops at the end, right? It can't jump that tiny gap to the next neuron.
That's right. The electrical signal triggers a chemical process at the synapse, that junction between neurons. This is where neurotransmitters come in.
The chemical messengers.
Exactly. When the action potential arrives at the axon terminal, it causes tiny sacs, vesicles filled with neurotransmitter molecules to release their contents into the synaptic gap.
So the message converts from electrical to chemical.
Precisely. These chemical messengers then float across that tiny gap and bind to specific receptor sites on the membrane of the next neuron, the receiving neuron. Tag the chemical messengers and communication basics.
And these receptor sites are specific, aren't they? It's not like any neurotransmitter can just dock anywhere.
Absolutely specific. It's often described using the lock and key mechanism. Each neurotransmitter molecule, the key, has a unique shape that allows it to bind only to a specific type of receptor site, the lock, on the receiving neuron.
So dopamine fits into dopamine receptors, serotonin into serotonin receptors, and so on. Ensures the message is precise.
Exactly. And when the neurotransmitter binds to its receptor, it causes a change in the receiving neuron, either making it more likely to fire its own action potential, an excitatory effect, or less likely to fire an inhibitory effect.
Okay, message delivered. But then what happens to those neurotransmitter molecules hanging out in the gap? They can't just stay there, right?
No, the synapse needs to be cleared quickly so the next signal can get through clearly. There are a few main ways this happens. The most common is reuptake.
Reuptake. The sending neuron takes them back.
Yeah, the axon terminal that released the neurotransmitters actually pumps them back inside, like recycling them.
Yeah.
They can then be repackaged into vesicles or broken down.
What if they're not taken back up?
Then they might be broken down right there in the synaptic gap by specialized enzymes. These enzymes chemically deactivate the neurotransmitter.
So either recycling or disposal keeps the signal clean.
Right. And there's another layer of control too. Neuromodulators. These are also chemicals, but they don't necessarily carry the primary message. Instead, they influence the sensitivity of the receiving neuron to the main neurotransmitters.
So they can turn the volume up or down on the conversation.
That's a good way to put it. They modulate or fine-tune the overall neural communication.
This whole chemical signaling process seems like a prime target for drugs to interfere with, right? Whether it's medication or recreational drugs.
Absolutely. Many drugs exert their effects by manipulating neurotransmitter systems at the synapse. We broadly categorize them as antagonists or agonists.
Antagonists. They antagonize, they block things.
Exactly. Antagonists are drugs that block the action of a neurotransmitter. Often they do this by binding to the receptor site without activating it, physically preventing the actual neurotransmitter from docking there, like putting gum in the lock.
So they reduce the effect of that neurotransmitter, and agonists do the opposite.
Agonists enhance the action of a neurotransmitter. They can do this in several ways. Some might mimic the neurotransmitter itself, binding to the receptor and activating it just like the real thing.
Like a master key.
Sort of. Others might work by increasing the amount of neurotransmitters available in the synapse, maybe by stimulating its release, or very commonly by blocking its reuptake.
Blocking reuptake. That keeps more of the neurotransmitter active in the gap for longer.
Precisely. Let's look at some examples our sources give. Caffeine, a pretty common stimulant.
My morning essential. How does that work as an agonist?
It primarily works by blocking the receptors for adenosine, a neurotransmitter that normally makes us feel drowsy. By blocking adenosine, caffeine indirectly increases alertness and the release of other stimulating neurotransmitters like glutamate.
Okay, so it blocks the sleepy signal. What about stronger stimulants like amphetamines or cocaine?
Those are powerful agonists primarily for dopamine, but also norepinephrine and serotonin. They work mainly by blocking the reuptake of these neurotransmitters, especially dopamine.
So they flood the synapse with dopamine by preventing it from being cleared away, leading to that intense euphoria and addiction potential.
Exactly. The reward pathway gets hijacked. Now, consider depressants like alcohol or anti-anxiety drugs, benzodiazepines like Valium or Xanax.
They calm things down. Are they antagonists then?
Actually, they're agonists, but for an inhibitory neurotransmitter called GABA, gamma-aminobutyric acid.
GABA. That's the main break neurotransmitter.
That's right. GABA's job is to reduce neuronal activity. Alcohol and benzodiazepines make the GABA receptors more sensitive, or they enhance GABA's effects, so they increase the inhibition in the brain.
Ah, so boosting the brake leads to the calming sedative effects. Makes sense.
It reduces anxiety, relaxes muscles, can cause drowsiness. And finally, think about antidepressants. Many common ones like SSRIs, selective serotonin reuptake inhibitors.
The name kind of gives it away. Reuptake inhibitors.
Exactly. They're agonists that specifically block the reuptake of serotonin, increasing its levels in the synapse. Others might target norepinephrine as well. The idea is to boost the signaling of these mood-regulating neurotransmitters. Hashtag key neurotransmitters and their clinical relevance.
Let's focus a bit more on dopamine. It seems to come up everywhere. Pleasure, movement, addiction, even serious mental illness.
It's incredibly versatile, involved in multiple brain circuits. Yes, it's famous for its role in the reward system feelings of pleasure, motivation, reinforcement learning. But it's also absolutely critical for controlling voluntary movement.
I've heard recently that the pleasure idea might be a bit simplified, that it's more about wanting or motivation.
That's a really important refinement. Modern research suggests dopamine is less about the pleasure of consuming a reward and more about the motivation, the anticipation, the seeking of the reward. It drives goal-directed behavior. It fires strongly when a cue predicts a reward is coming.
Okay, the wanting system, not just the liking system. And when dopamine signaling goes wrong, the consequences are huge.
Definitely. Take schizophrenia. While it's very complex, one leading hypothesis involves hyperactivity in certain dopamine pathways, which might contribute to symptoms like hallucinations and delusions. Too much dopamine activity in the wrong places.
And the flip side, too little dopamine.
That leads directly to Parkinson's disease. In Parkinson's, the specific neurons that produce dopamine in a part of the brainstem called the substantia nigra die off. These neurons normally project to the basal ganglia, which control movement.
So losing that dopamine input messes up movement control.
Severely. It causes the classic symptoms: tremors, muscle rigidity, slowness of movement, difficulty initiating actions, balance problems. It's a devastating illustration of how crucial one neurotransmitter is for a fundamental function.
Wow. Okay, one more key group before we move to the brain itself. Endorphins, the body's natural painkillers.
Exactly. Endorphins are inhibitory neurotransmitters that function similarly to opioid drugs like morphine. They're released in response to pain, stress, but also strenuous exercise.
Or runner's high.
That's thought to be partly due to endorphins. They inhibit pain signals and can produce feelings of euphoria or well-being. It's an adaptive mechanism that helps us push through pain or injury in an emergency or cope with prolonged exertion.
Okay, we've built the system from the neuron up. Now let's get to the main event. The brain itself. It's incredibly complex, but our sources organize it sort of evolutionarily into three main layers, right?
That's a helpful way to approach it, starting with the oldest, most fundamental part, the hindbrain. It sits at the base of the skull, connecting to the spinal cord.
And it handles the really basic survival stuff.
Absolutely. Structures here, control functions essential for life itself. You've got the medulla.
Medulla. Sounds important.
It is. Damage here is often fatal. The medulla regulates core automatic functions: heartbeat, breathing, blood pressure, swallowing, coughing. Things you never think about but can't live without.
Okay. What else is in the hindbrain?
There's the pons, which sits just above the medulla. Pons means bridge in Latin. It acts as a relay station, passing information between the cerebellum and the rest of the brain. It's also involved in regulating sleep and wakefulness cycles.
And the third hindbrain structure, the cerebellum, the little brain.
Right, the cerebellum, tucked away at the back. It looks like a miniature version of the main brain hemispheres. Its primary, well-known role is coordinating voluntary movements, posture, and balance.
So walking smoothly, reaching accurately, playing an instrument that relies heavily on the cerebellum.
Critically, damage here leads to jerky, uncoordinated movements, problems with balance, difficulty with fine motor skills. We're also learning it's involved in more complex things, like learning motor skills and even some aspects of attention and language.
Okay, so hindbrain equals survival, basic movement coordination. Moving up, we get to the midbrain.
Yes, a relatively small section connecting the hindbrain to the forebrain above it. It acts as another relay station, particularly for auditory and visual information, and it plays a role in controlling eye movements.
Is there a key structure in the midbrain we should know?
A really important network runs through the midbrain, and also parts of the hindbrain is called the reticular formation, sometimes called the reticular activating system.
Reticular formation? Sounds like a net.
It is. It's a network of neurons. Its crucial role is regulating arousal, alertness, and attention. It acts like a filter for incoming sensory information, deciding what's important enough to bring to conscious awareness.
So it helps you wake up in the morning, stay focused, and filters out background noise.
Exactly. Damage here can lead to problems with consciousness, even coma. It's the brain's attention dial and gatekeepers.
Right, hindbrain, midbrain. Now the biggest, most evolutionarily recent part, the forebrain. This is where the really complex human stuff happens, right? Thought, language, personality.
Absolutely. The forebrain is the largest and most complex part. It includes the two large cerebral hemispheres that everyone visualizes when they think of the brain, plus several crucial structures tucked underneath them.
Let's start with those structures underneath. There's the thalamus.
Right. The thalamus is often called the brain's sensory relay station, or switchboard. Almost all sensory information, sight, sound, taste, touch, but not smell, goes through the thalamus first.
So it receives the signals and directs them to the right processing areas in the outer brain.
Exactly. It sorts the signals and sends them on to the appropriate regions of the cerebral cortex for higher level processing. It also plays a role in regulating sleep and wakefulness.
Okay, sensory gateway. What about the hypothalamus? Hypo meaning below the thalamus.
Correct. It's small, just below the thalamus, but incredibly powerful. The hypothalamus is the master regulator of many fundamental drives and bodily functions needed for survival.
Like what specifically?
It controls body temperature, hunger, thirst, fatigue, sleep-wake cycles. It's also a key link between the nervous system and the endocrine system, the hormone system, influencing the pituitary gland. And it plays a major role in emotional responses and motivated behaviors, often summarized as the 4 Fs.
Fighting, fleeing, feeding, and mating.
You got it. Basic survival drives. Then there's the basal ganglia.
Basal ganglia. Sounds like a cluster of cells.
It is a group of nuclei deep within the forebrain, crucial for regulating voluntary movement, particularly initiating and smoothing out muscle movements. We mentioned Parkinson's disease earlier that involves the death of dopamine neurons that project to the basal ganglia, disrupting its function.
Ah, okay, so it's central to movement control. And finally, the limbic system. This is the emotional brain.
It's heavily involved in emotion, yes, but also memory. The limbic system isn't one single structure, but a collection of interconnected structures looping around the edge of the thalamus and the hypothalamus. Key players include the hippocampus, the amygdala, and parts of the hypothalamus and thalamus.
Hippocampus. That's memory, right?
Crucial for forming new long-term memories, particularly memories for facts and events. The amygdala, right next door, is central to processing emotions, especially fear and aggression, and linking emotions to memories.
So the limbic system connects our feelings to our experiences and helps store them. That's why emotional events are often remembered so vividly.
Precisely. It integrates emotion, motivation, and memory.
Okay, now let's zoom out to the main part of the forebrain, the two big cerebral hemispheres. They look pretty symmetrical, but they aren't identical in function, are they? This is lateralization.
Exactly. Lateralization refers to the division of labor, the specialization of function between the right and left hemispheres. While they constantly communicate and work together, each hemisphere tends to dominate in certain types of tasks. And generally, each hemisphere controls the opposite side of the body.
So the left brain controls the right hand, right eye, etc. And the left hemisphere is usually the language brain, the analytical one.
In most people, yes, especially right-handers. The left hemisphere is typically dominant for language ability, speaking, understanding language, reading, writing. It's also generally better at logical analysis, breaking down problems, mathematical calculations, sequential processing.
The detail-oriented side. What about the right hemisphere?
The right hemisphere tends to excel at nonverbal processing. Things like understanding spatial relationships, navigating, reading maps, recognizing geometric shapes. It's also generally better at recognizing faces, interpreting emotional expressions in faces and voices, appreciating music and art.
So more holistic, big picture, creative, and emotional processing.
That's a good summary. It synthesizes information, grasps overall patterns, and handles the nuances of social and emotional cues.
How do these two specialized halves talk to each other? They need to coordinate, right?
Absolutely. They're connected by a massive bundle of nerve fibers called the corpus callosum. Think of it as a superhighway, allowing constant, rapid communication between the two hemispheres, integrating their specialized abilities.
And we learned a lot about this from split-brain patients, didn't we? People who had the corpus callosum cut, usually for epilepsy.
Those studies were revolutionary. They really highlighted the separate abilities of each hemisphere when communication was severed. For example, if you showed an object only to their left visual field, which goes to the right hemisphere. They couldn't name the object because language is usually in the left.
But they can maybe pick it up with their left hand?
Exactly. Their left hand, controlled by the right hemisphere, could identify the object by touch or point to a picture of it. It showed the right brain knew it was, it just couldn't verbalize it. Really fascinating stuff.
And speaking of language being lateralized, there are specific areas in the left hemisphere critical for it, Broca's and Wernicke's area.
Yes, two key language centers, usually in the left hemisphere. Broca's area, located in the left frontal lobe, is crucial for speech production. Damage here, Broca's aphasia, leads to difficulty forming words, speaking haltingly, though comprehension might be relatively intact.
So they know what they want to say but can't get it out fluently.
Right. Wernicke's area in the left temporal lobe is critical for language comprehension. Damage here, Wernicke's aphasia, results in difficulty understanding spoken and written language. They might speak fluently, but the words can be jumbled, nonsensical, like a word salad, because they aren't processing the meaning correctly. Hashtag brain adaptability and handedness.
So we have this incredibly complex, specialized structure, but it's not set in stone from birth, is it? The brain can change. This is plasticity.
Yes, and this is one of the most exciting areas of neuroscience. Plasticity refers to the brain's remarkable ability to change its structure and function in response to experience, learning, or following injury. It can reorganize itself, form new neural connections, strengthen existing ones.
So it's not a fixed machine, it's constantly adapting.
Throughout life, yes. We see it after brain damage, like a stroke, where undamaged areas might gradually take over some of the functions of the injured part. We also see it with learning.
Like the London taxi driver study.
Exactly. Studies show that experienced London taxi drivers who have to memorize the incredibly complex map of London actually have physically larger hippocampine, the brain region crucial for spatial memory compared to control groups. Their brains structurally change due to their intensive learning and experience.
Amazing. It really highlights that the brain is dynamic. Okay, one last point related to specialization. Handedness. Most people are right-handed. Is that linked to brain lateralization?
It is. About 90% of people are right-handed, which typically corresponds with the left hemisphere being dominant for fine motor control as well as language. Hand preference seems to develop very early, possibly even before birth.
Is it purely genetic?
Genetics certainly plays a role, but it's not the whole story. Environmental factors likely contribute too. It's complex. Interestingly, men are slightly more likely to be left-handed than women, though the reasons for that aren't fully clear.
We've spent a lot of time on the nervous system, fast, electrical, targeted communication. But there's another major communication system in the body, right? The endocrine system. Slower, more widespread.
Exactly. The endocrine system uses chemical messengers called hormones. These are secreted by glands directly into the bloodstream.
Not across synapses, but into the blood.
Right, so hormones travel throughout the body via the circulatory system. This means their effects are generally slower to start than neurotransmitters, but they can be much longer lasting and affect many target organs simultaneously.
And its main job is also maintaining balance, homeostasis.
Yes, maintaining that stable internal environment is a key function. The endocrine system regulates growth, metabolism, sexual development and function, mood, sleep, and responses to stress over longer timescales.
What are the major glands involved? Our sources list quite a few.
The key players include the pineal gland, melatonin, sleep, the hypothalamus, which controls the pituitary gland, the master gland influencing others, the thyroid gland, metabolism, the adrenal glands, stress hormones like cortisol and adrenaline, the pancreas, insulin and glucagon for blood sugar, and the gonads, ovaries in females, estrogen, progesterone, and testes in males, testosterone.
That's quite a network. The pancreas and blood sugar example seems like a classic case of homeostasis in action.
It's a perfect example of a negative feedback loop. Let's say you eat something sugary and your blood glucose level rises.
Sugar rush.
The pancreas detects this increase. In response, specialized cells in the pancreas release the hormone insulin into the bloodstream.
And what does insulin do?
Insulin acts like a key, signaling cells throughout your body, especially muscle, fat, and liver cells, to take up glucose from the blood to use for energy or store for later.
So it pulls the sugar out of the blood, lowering the level.
Exactly. As blood glucose levels fall back towards the normal range, the pancreas detects this and reduces its insulin secretion. It's a self-regulating system to keep blood sugar within a tight window. Tag hormones and behavior.
Now, the connection between these hormones and our behavior, our psychology, is fascinating. It seems to go both ways. Hormones affect behavior, but behavior can affect hormones too.
Absolutely reciprocal. Take testosterone, the primary male sex hormone, though females have it too in smaller amounts. It's essential for development, but variations in levels are linked to certain behaviors.
Like aggression.
It's more complex than a direct cause, but higher testosterone levels are associated with increased dominance, competitiveness, risk-taking, and sometimes aggression, especially in response to social challenges. We see extreme effects with anabolic steroid abuse, which involves ingesting synthetic testosterone and is strongly linked to increased irritability and aggression, roid rage.
Okay. What about oxytocin, often called the love hormone or cuddle chemical?
Oxytocin plays a huge role in social bonding, trust, and empathy. It's released during childbirth and nursing, facilitating mother-infant bonding. It's also released during positive social interactions, like hugging or sex, promoting pair bonding and feelings of connection.
So it's purely positive?
Not necessarily simple. Research suggests it primarily promotes bonding and trust within your own group, your in-group. It might even increase defensiveness or suspicion towards perceived out-groups. And interestingly, it's also linked to increased conformity to your group's norms.
Conformity. Huh. So it makes you more likely to go along with your buddies.
It seems to enhance the desire for social connection and acceptance within the group, which can manifest as conformity. It's not just love, it's about complex social navigation.
Fascinating. And the thyroid hormones, they seem directly linked to mood and energy.
Very directly. The thyroid gland controls the body's metabolic rate. If it produces too much hormone, hyperthyroidism, metabolism speeds up. People might feel anxious, irritable, restless, have trouble sleeping, lose weight.
Wired and too little.
Hypothyroidism slows metabolism down. That can lead to fatigue, sluggishness, weight gain, feeling cold, and even depression. It's a clear example of how purely physiological hormone levels profoundly impact psychological state.
Understanding all this, the structures, the chemicals, the hormones, requires some sophisticated tools. How do scientists actually study the brain in action? There are non-invasive ways, right? Looking from the outside.
Yes, several powerful imaging techniques let us see brain structure and activity without surgery. The EEG, electroencephalograph, is one of the oldest.
That's the one with electrodes on the scalp, measuring brain waves.
Exactly. It records the overall electrical activity of large groups of neurons firing. It has excellent temporal resolution, meaning it tells you when brain activity changes, millisecond by millisecond. Great for studying sleep stages, detecting seizures, or tracking rapid responses to stimuli.
But it doesn't show where the activity is happening very precisely.
No, its spatial resolution is poor. For structure, we use scans like CT, computed tomography, or MRI, magnetic resonance imaging.
CT scans. Those are like fancy x-rays.
Sort of. A CT scan takes multiple X-ray images from different angles and uses a computer to create cross-sectional slices of the brain. It's good for seeing basic structure, identifying tumors, strokes, or brain injuries quickly.
And MRI, magnetic resonance.
MRI uses strong magnetic fields and radio waves, not X-rays. It provides much more detailed images of the brain's soft tissues than CT. You get really clear pictures of brain structures. It has excellent spatial resolution.
Okay, so EEG for when, CT/MRI for what structure? What if we want to see which parts are active during a task? Functional imaging.
That's where PET, positron emission tomography, and fMRI, functional MRI, come in. A PE scan involves injecting a small amount of a radioactive tracer, usually attached to glucose, sugar.
Brain cells use glucose for energy.
Exactly. The most active brain areas use the most glucose. The PE scanner detects the radioactivity, creating a map showing which parts of the brain are working hardest during a specific task, like reading or problem solving. It shows function, but the temporal resolution isn't great. fMRI is often preferred now as it also measures activity by tracking blood flow changes, which correlate with neural activity, but without radioactivity, and often with better spatial and temporal resolution than PET.
Right. Now, there are also more invasive methods, used mainly in animal research or sometimes during necessary brain surgery.
Yes, these allow for more direct manipulation and recording. Lesioning involves precisely damaging a specific brain area in animals to observe the effect on behavior, helping determine that area's function.
Understand function by seeing what happens when it's broken.
Basically, electrical recording uses microelectrodes implanted directly into the brain to record the activity of individual neurons or small groups while the animal performs tasks. Very precise information.
And you can stimulate too, right?
Yes. Electrical stimulation involves using weak electric currents to activate specific brain areas and observing the resulting behavior. Famously used by Penfield during brain surgery on conscious patients to map out motor and sensory areas.
Okay, we've covered the structure, the wiring, the chemistry, but the blueprint for all of that comes from our genes, our heredity. This brings us to the classic and often tricky question. How much of who we are, our traits, our behaviors, our risks for disorders comes from nature, genes, versus nurture, environment?
It's one of the fundamental questions in psychology and biology, and it's incredibly challenging to disentangle because for humans, genes and environment are always interacting. We can't ethically assign people different genes or completely control their environments.
So researchers have clever ways to estimate the relative influence, using natural experiments, basically, like kinship studies.
Exactly. Kinship studies leverage the fact that biological relatives share predictable amounts of genetic material. The simplest type is familial association studies.
That just looks at whether a trait runs in families.
Pretty much. Researchers examine how often a trait or disorder, say depression or musical talent, appears among close relatives compared to more distant ones or the general population. If it clusters in families, it suggests a genetic link.
But the big problem there is that families share environments too, right? Same house, same diet, same upbringing styles.
Precisely. Familial studies can't separate shared genes from shared environments. That's why twin studies are so powerful.
Comparing identical versus fraternal twins.
Right. Identical monozygotic twins develop from a single fertilized egg splitting in two, so they share 100% of their genes. Fraternal dizygotic twins develop from two separate eggs fertilized by two different sperm, so they're genetically like regular siblings, sharing about 50% of their genes on average.
But both types of twins typically grow up in the same family environment at the same time.
That's the key assumption. So if identical twins are significantly more similar, have a higher concordance rate for a particular trait like IQ, personality trait, or risk for schizophrenia than fraternal twins are, it provides strong evidence for a genetic contribution to that trait.
Because the main difference between the two groups is the degree of genetic similarity, not the environment.
Exactly. The difference in concordance rates gives an estimate of heritability.
But even identical twins might have slightly different experiences. To really separate genes and environment, researchers look at adoption, right? Adoptee studies.
Yes, adoptee studies provide another crucial angle. They compare adoptees to both their biological parents, who provided their genes, and their adoptive parents, who provided their upbringing environment.
So if an adoptee is more similar on a trait, say introversion, to their biological parents whom they never knew, than to their adoptive parents who raised them.
That points strongly towards genetic influence. Major. Conversely, if the adoptee resembles their adoptive parents more than their biological parents on that trait, it points towards environmental influence, nurture.
By comparing these patterns across many adoptees, researchers can estimate the relative contributions of genetics and environment to various traits and disorders. It's how we get those heritability estimates you often hear about.
Right. And it's important to remember heritability is a population statistic, telling us about variation within a group, not about how much of one individual's trait is genetic. And even highly heritable traits are often still significantly influenced by environment. It's almost always an interaction. ##8. Conclusion and provocative outro.
Wow, okay, that was a deep dive indeed. We've gone from the single neuron firing, that action potential jumping down a myelinated axon,
To the chemical ballet of neurotransmitters like dopamine at the synapse, influenced by agonists and antagonists,
up through the major divisions of the nervous system, the CNS and PNS, the constant push and pull of the sympathetic and parasympathetic systems.
and into the incredibly complex structures of the brain. The survival functions of the hindbrain, the arousal regulation of the midbrain's reticular formation, the complex thought and emotion hubs in the forebrain, like the thalamus, hypothalamus, limbic system.
Not forgetting the specialized hemispheres, the language centers, and the body's other communication network, the endocrine system with its hormones.
And tying it all together was that recurring theme. This isn't just fixed hardware. The brain's plasticity means it's constantly changing, adapting, reorganizing based on everything we experience.
Which brings us right back to that nature-nurture interaction we just discussed. The genetic blueprint provides the starting point, the potential.
But the environment, our experiences, learning, even our own thoughts, constantly shape how that blueprint unfolds, physically altering the brain's connections and chemical balance through plasticity. It's a continuous feedback loop.
It really drives home that we're dynamic systems, not static machines. Which leads us to a final thought, something for you, our listeners, to chew on.
Yeah, consider this. We know the brain has this incredible capacity for plasticity, where functions can shift and reorganize, especially after damage or with intensive learning. We also know that our internal chemistry, hormones like oxytocin or stress hormones, profoundly shapes our social behaviors, our trust, our empathy, even our tendency to conform.
So,
So how much of what you think of as your fixed personality, your unchangeable self, is truly permanent? How much of it is actually just the current state, perhaps a temporary state of your specific neural connections and your fluctuating chemical messengers, all of which are adaptable and responsive?
are we less fixed than we think?
Perhaps. The you of today is biologically different from the you of last year and will be different again next year because your brain is constantly learning, adapting, rewiring. Maybe personality is less of a static thing and more of an ongoing process shaped by the dynamic interplay of your biology and your life. Something to think about.